First of all, what is DNA microarray? Microarray is a rectangular grid of spots printed on a glass microscope slide where each spot contains DNA for a different gene. DNA microarray allows us to study thousands of genes within a genome at one time by combining robotics, chemistry, computer science, and biology. Every spot on the chip represents a different coding sequence from different genes. Each spot on the chip is made of a DNA probe that can pair with the cDNA that was created. In general, DNA microarray is used to determine the expression level of genes and investigate the differences between a healthy cell and cancer cell. This figure shows how DNA microarray works in general. In detail, DNA microarray works in 7 different steps. We start with sample collections. We need to obtain cells with genes that are needed for analysis. The next step will be mRNA isolation. We need to extract the RNA from the sample by using column or solvent. After that, isolate mRNA from the rRNA and tRNA. Since mRNA has a poly-A tail, so we can use a column containing bits with poly-T tails to bind with the mRNA. After that, we need to rinse with buffer to release mRNA from the bits. The next step will be preparation of labeled cDNA. A labeling mix is added to the RNA. A labeling mix is actually a mixture containing poly-T primer, reverse transcriptase, and fluorescently dyed nucleotide. After that, we need to label the target with fluorophore. For example, the commonly used fluorophore is CY5, which are red in color, and CY3, which are green in color. Primer and reverse transcriptase will first bind to the mRNA, and then we need to add fluorescently dyed nucleotide to create a complementary strand of DNA. The next step will be hybridization. We need to apply the created cDNA to the microarray plant. If we want to compare two samples, we need to apply both samples to the same plant. cDNA will bind to the single-strand DNA that already present on the plant. After that, we need to wash off the unbound cDNA to see what has been bound to the microarray. The last 2 steps will be visualization and data analysis. In order to visualize the bound cDNA, we need to place the slide with the microarray chips inside a dark box. And then we need to scan with a high-resolution laser that will detect the bound fluorescent label. The information and images were then sent to the computer for analysis. For data analysis, we need to create a ratio image by using computer to calculate the ratio of intensity. The ratio image might contain 4 different colors of spot. Green spot indicates that the gene was strongly repressed. Red spot indicates that the gene was strongly expressed. Yellow spot indicates that the gene was neither strongly expressed nor strongly repressed, which means that the genes are inactive. Black spot indicates that none of the cDNA has bonded to the DNA in the gene located in that spot. Besides analyze the data by using a ratio image, we can also analyze the data by using a scatter plot.
Applications of microarray. Gene expression analysis used in the comparison of gene transcription level in clinical conditions such as identify diagnostic or prognostic biomarker, classify disease, monitor the response to therapy, understand the mechanism involved in the genesis of disease process. Only a small amount of starting RNA is needed. Transcription factor binding analysis. Chip is a combination with chromatin immunoprecipitation. It is known as chromatin immunoprecipitation on the chips or microarray. The purpose is to determine the binding sites of transcription factor. Transcription factors cross-link to DNA with formaldehyde. DNA is fragmented. DNA-bounded TF is purified with antibody or by affinity chromatography. DNA is released from the TFs and proceed to amplification, labeling, and hybridization to the array. Cancer research. Microarray can help in identification of single nucleotide polymorphism, SMP, and mutation, tumor classification, identification in cancer biomarker, and identification in target genes of tumor suppressor. Wide range genetic abnormality of tumors, for example, breast carcinoma, lymphoma, and bladder carcinoma, can be mapped. Antibiotic treatment. Antibiotic resistance has become a global health problem. Bacterial genomic DNA often outlasts the viability of the bacteria. Diagnosis can be made using a small amount of DNA as opposed to the large number of bacteria needed for culture. The test specificity, sensitivity, and the predicted value of positive and negative tests will be calculated.
Here we end our presentation. Thank you.
Express sequence tag or EST is a short fragment of mRNA sequence derived through single sequencing reaction performed on clones from cDNA libraries that can identify gene transcript. An EST is a sequence tag site that is detectable by PCR, which can be served as a landmark in the physical map of the genome. To produce EST, the exons of mRNA are transcribed as part of the heterogeneous nucleic RNAs in vivo and are subsequently spliced together to form mRNA. In vitro, reversal of this process is possible by reverse transcription of the complete set of mRNAs. This yields cDNA libraries that correspond to the gene expressed in that sample. Sequencing a short fragment, probably less than 100 bases at one or both ends of each cDNA produce a tag, so-called EST, which has enough information to identify the expressed gene. Sequencing only the beginning portion of the cDNA will produce 5' EST. 5' EST is obtained from the portion of a transcript that usually code for a protein because these regions tend to be conserved across species and do not change a lot within a gene family. Sequencing the ending portion of the cDNA will produce 3' EST. 3' EST are likely to fall within non-coding or untranslated region, and therefore tend to exhibit less cross-species conservation than the coding sequence do. This flowchart will show the preparation of a cDNA libraries in a plasmid. The mRNA will be transcribed into a stable, double-stranded cDNA that is protected by methylation. Then the cDNA will clone into a plasmid libraries. Next, the cDNAs will be sequenced at each end to obtain EST. The EST will be compared to one another and to the genomic sequence. Then the tentative unique genes can be obtained. cDNA can be directly expressed in prokaryotic system, which means the eukaryotic genes can be expressed in prokaryotes that helps in the transcription process of prokaryotes. Large-scale sequencing is required to get statistical significance. In the cDNA libraries, 3' ends are heavily represented because most are primed with oligo. cDNA of protein can facilitate to generate antibodies and monoclonal antibodies. Then cDNA can be used in the study expression of mRNA.
So now, what can EST tells you? There are 3 examples of application here, which is firstly gene discovery, second sources as genetic marker, and thirdly it can be used in plant hybridization experiment. But how does this related with EST? Here we have to know that EST represents a particular type of sequence tag size useful for the physical mapping of genomes. In many genome projects, EST is widely used as application, as novel genes can be found by querying the EST database with a protein or DNA sequence. For example, there is a new member found in human Ly6 family through EST sequencing, and 66 human EST were identified and mapped based on their resemblance to 66 Drosophila genes. That's the end of our presentation. Thank you.